In the first part of the lecture, we will look on transitions. The particular transition we are interested in is that of protein folding, where proteins go from a folded state to an unfolded state and reverse. Now, this process is very obvious right at the beginning of the birth of a protein. The protein is read up from the ribozyme, comes out as a string of amino acids, and those have to fold into a structure that is giving the function to a protein. The question is, how does this work, and how stable is this, and where does the folding depend on? At the very end of this first part, we will also get back to the observations of the force extension of DNA, where it has been reported that at 65 piconewton there's a dramatic change happening. That we will be able to explain by the models that we develop for protein folding. As mentioned, it's not surprising that proteins change their shape. Right at the beginning, they have to fold into the right. Yet what came to a surprise, and that has been shown by Ann Vincent for the first time, that proteins denature. So they unfold and refold again. So this dynamic process was a big surprise at the time. And what has been done in these experiments is that Ann Vincent looked for the amount of folded protein, then added a denaturant, which in his case was urea, something that dissolves hydrogen bridges, and then unfolded the protein, then removed urea again and looked for the refolding. And the refolding you actually see in this experiment here. So the take-home message is that a folded protein is not stable. It can be unstable, and even this being stable and unstable could be the function of the protein. That has been shown, for example, for a protein which is found in your muscle, titin, which functions as a shock absorber for the action of muscles and prevents that you rupture muscle cells by contracting your muscle. Let's briefly discuss first how Ann Vincent distinguished folded from unfolded proteins. And for that he used an optical technique. A monochromatic and linearly polarized light source was projected through a sample of length d. Now, the sample contained a watery solution and proteins at a given concentration, small c. Then, after the light has passed through this sample volume, an analyzer was placed, and what you could observe is that there's a change of rotation of the polarization of the light. Now you see a change in polarization of light when the sample contains non-symmetric objects like helices in a protein. So what you could analyze here is the mean rotation angle, mean theta. If the sample would contain 100% of unfolded protein, then the change of rotation per protein would be randomly oriented, and the mean orientation or turning of the polarization is 0. Yet for folded proteins, because of helicity in those folded proteins, that does not hold, and the mean polarization would be finite. Now let's look on a different experimental realization as compared to that of Ann Vincent's experiment. Here you see how the mean polarization in a sample is changed as a function of the temperature. At low temperature, At low temperature, you have the situation that the mean polarization angle is small, and at high temperature, it even turns sign and goes to a higher value. Whereas for the random coil, so this is a protein that does not fold at all, the polarization will just stay and is independent of the temperature. Now let's make up a model that could explain these experimental results. And the model will include a two-state model, something that we saw already in last week's lecture. We assume that the protein is made up of n amino acids. And we assume that we can assign any amino acid, AAI, to be either in the folded state, characterized by a sigma I of +1, or in an unfolded state, where sigma I is -1. Now for folded, we assume that it's in a helical state, where for unfolded, it's called in the coiled state, which is just a random polymer. For the helical state, we know that the helix is defined by interactions of amino acid I with amino acid I plus 4. So there is an additional constraint, namely that if the amino acid I has a sigma I of +1, then amino acid I plus 4 must have a sigma or I plus 4 of also +1. Now, from these model assumptions, we can now calculate the total optical rotation of the system, capital theta, which is just the number of amino acids that are in the helical state and helix times small theta, which just is a number telling me that if an amino acid adds to a helix, it will turn the polarization by a certain amount. Now that we can rewrite in terms of the sigma i's, and we find that theta is equal to 1/2 times 1 plus 1 divided by n, sum on all i's, sigma I times small theta, where the sum on all sigma i's divided by n is the mean sigma of the system. Now the next step, we would like to develop a stat-mech model to understand the experimental findings. For that, we now develop a model for the change in the free energy on adding one amino acid to a helical structure. So here we consider the free energy delta GI as given by delta epsilon I, internal energy by releasing or obtaining energy, minus T delta S. Now adding an amino acid to a helical structure requires a hydrogen bond formed between the amino acid I and the amino acid I plus 4. Now formation of this bond will lead to a release of energy. Yet the hydrogen bonds to the surrounding water molecules will have to be broken. Now adding all this up, typically this means that the formation of a helix will need energy, and delta epsilon I is larger than 0. Now the second part of the free energy is the entropic part, which is delta Si. That has two contributions. First is formation of a helix will decrease the entropy of that particular amino acid. So delta SAA is negative. Yet at the same time, there's a breakage of hydrogen bonds towards the environment, the water, leading to an increase of the entropy of the water, as we explained in the hydrophobic effect. So in total, delta Si is positive. Now it's handy to look on yet another expression that we obtained from the above considerations, and that is delta GI divided by KBT. Now filling in our definition of delta GI, which equals to delta epsilon I minus T delta SI, we can reformulate this expression and finally obtaining that delta GI divided by KBT is equal to delta epsilon I divided by KB times Tm minus T divided by T times Tm, where we define the Tm being the ratio of delta Si divided by epsilon I. Tm is a temperature, and sometimes it's called the mid-temperature, or in protein folding, the melting temperature of the protein. So far we looked on the energy that is associated with the addition of one amino acid. We also realized earlier that in order to form a helix, it is required that there is some interaction between the amino acid I and the amino acid I plus 4. So the idea is that we now propagate this interaction from amino acid I to amino acid I plus 1, and that interacts again to amino acid I plus 2, and so forth to amino acid I plus 4. Now the assumption that we take is that this interaction is purely entropic, meaning that the interaction energy is equal to minus T delta S helix, which is the loss of the entropy because we have to fix for those amino acids, those 5 amino acids, the angles that are allowed. With this in mind, we easily can estimate this interaction energy, and our estimation would be that this is equal to T times Kb log of 3 to the power 2, where the 3 counts the number of amino acids between the amino acid I and I plus 4, and the 2 refers to the 2 angles that we have to fix for those amino acids. Finally, now we derive to the total energy if we change the location of amino acids from the coil state into a helical state, which we now call delta E, the total energy change for the whole chain, is equal to the sum on all I for all units amino acids of the chain, delta epsilon I minus T delta Si times sigma I, which is the energetic part, plus this interaction part that we just introduced, gamma times sigma I plus 1. Or rewriting this in terms of the melting temperature is equal to the sum on all I, epsilon I, 1 minus T divided by Tm times sigma I plus gamma sigma I plus 1. So what did we achieve so far? Well, we modeled the transition from helix to coil in the protein by a two-state model, where there is a change of the free energy G from the folded to the unfolded state. The different contributions we also at least estimated, and we found that delta G divided by KBT is equal to delta E divided by KB, T melting minus T divided by T melt. Now looking back to the last lecture, there we looked on unfolding of protein or the DNA. And again, we found that we could nicely describe this process by a two-state model where we had the polymer either in a stretched or in an unstretched way. And there the energy difference between the stretched and unstretched was equal to FL divided by KBT. Further in our description, we needed to introduce an interaction parameter between neighboring amino acids For the description of the requirement for a helix formation, and that we equaled here is equal to gamma divided by kBt equals to T times delta S helix divided by kBt. Likewise, we had to introduce an interaction parameter, also gamma divided by kBt, that describes interactions between neighboring nucleotides in the DNA. Now, further, our definitions allowed us to calculate the mean rotation. The mean of capital theta is equal to theta times the mean of sigma. And likewise for the extension experiments, where the mean extension, mean z, is equal to L times the mean of sigma. Now you see the advantage of such very general modeling that we did. So we easily can associate the different parameters in the Hamiltonian or in the energy. And the association would be that, the addition of the amino acids, which is equal to delta G divided by kT, we can associate with the stretching energy equaling F times L divided by kBt. Likewise, we have the same interaction parameter, so delta S helix times T divided by kBt is equal to gamma divided by kBt, and the mean rotation we can associate with the mean extension. Hence, we now use the result that we obtained for the stretching experiments of last week and calculate the mean rotation angle that reports on the folding. And what we obtain is that the mean rotation angle divided by the instantaneous rotation angle is equal to sine hyperbolic delta G divided by kBt divided by square root sine hyperbolic squared delta G divided by kBt plus e to the power minus gamma divided by kBt. Further, we have to add some calibration factors C1 and C2, where C1 displays an offset in the rotation, and C2 is the scaling factor that describes our observations. Now, taking this description, the theoretical curve, we now can fit the data points to what we see, and we obtain the respective parameters, for example, the melting temperature for that particular protein being 285 Kelvin, and the interaction parameter, which the fit tells us is 2.2, where we estimated should be log 9, which equals 2.1. So let's summarize what we obtained. So we obtained a description using the Ising model for the two-state folding problem of a protein. And the important parameters were given by the melting temperature that is associated to the change of the delta G from the folded to the unfolded state, and an interaction parameter. Now, what we would like to understand now is how does the interaction parameter really change the curve that we obtain. Now, in order to do so, we look for the steepness at the inflection point of that curve. The inflection point is at the place where the temperature approaches the melting temperature. So what we have to calculate is what is the derivative of the mean rotation angle with respect to the temperature. Now, we have terms of sine hyperbolic in this equation that just changes to the cosine hyperbolic divided by through the square root, and then we have to take the derivative of the square root, which gives us another cosine hyperbolic as an internal derivative times the sine hyperbolic, again, of the internal derivative, times the internal derivative of the argument of the sine hyperbolic, which is delta E divided by kb t squared. Now, we're not very interested in the full functional form, but only interested in at the inflection point, where for the inflection point the cosine hyperbolic is equal to 1, the sine hyperbolic is equal to 0, and the temperature is equal to the melting temperature. Now, using these relations, we obtain that the steepness at the inflection point is given by C2 times delta E divided by kBt melting squared times e to the power 2 gamma. Hence, it's proportional to e to the power 2 gamma. And this gives now an interpretation of the effect of the interaction. By increasing the interaction, the steepness of the curve is increasing. And this is exactly what cooperativity is about. It will lead to an increase in steepness of transitions as to nonlinearities that we introduce. So as a summary, so what did we achieve? We looked for the helix-to-coil transition and looked on how a protein is folding as a function of temperature. And that we analyzed in terms of the optical rotation that we can see in a cuvette. By mapping the problem to the Ising model, we could read off the solution, and we found that it nicely explains the data by this S-shaped curve. Further, we realized that the steepness of the curve at the inflection point is determined by the interaction parameter. Now, further experiments have been done, and the experiment that we discussed so far was a very long polymer or a very long protein. The question would now be if we go to shorter polymers. Having shorter polymers, you see that the curves drastically change. Reducing the amount of polymer units from 1500 to 46, we see that the curve largely flattens. And for 26, it even flattens further. Now, that is something we would not expect, because we know that the steepness is given by the interaction parameter. Yet, what we have to recall is that the partition function that we have been using in our description here was the partition function for very long polymers. Since the partition function is given by the sum on the two eigenvalues of the transfer matrix to the power n, lambda plus to the power n plus lambda minus to the power n, we assumed at some time that for very long chains, the minus solution just disappears. But what you see here is that for a restricted number of polymer units, that it doesn't hold anymore, and we have to find more complex solutions. Now we also can turn back to the force extension curves of stretching of the DNA. We found in the last lecture that the initial phase in the low force regime we could explain using the freely jointed chain or the interacting chain model. Yet at 65 piconewtons, something dramatic happens. Well, what is happening here is actually a transition of the structure of DNA. And that is called the B to Z transition, where the B form is the regular B form, where we have left-handed helix as explained. And at 65 piconewton, we pull that hard that we break all the interactions between the nucleotides, and the nucleotides actually will flip to the outside with the backbone in the middle and the nucleotides or the bases pointing outward. That is the so-called the Z form, which had been proposed by Pauling before it was known that the DNA had the B form.